Although this is all based on scientific research, I'm trying to present this in a way that just the people who just love oaks can understand. So hopefully I'm not going to talk about PCRs in too much detail, and if I'm getting too scientific, let me know and I'll stop and I'll make it understandable to most people. What I want to talk about is tell you the story about this one shrub oak in West Texas called Quercus hinckleyi. And I'll start off by giving you the background of the species, some of the details of the species, and then conservation issues that are related to it. And then I have to talk about the genetic study. I'm not going to talk about the details of it. I'm going to put up at the very end of the talk a couple of papers that we've written if you really want to get into that much detail on it. And then how, in addition to answering some conservation questions, we looked at broader implications. Some assumptions that are made about rare plant species, and then just leave you with some key thoughts. So that's actually Cornelius Muller, which we've heard about a couple times here, either species names or otherwise, very famous botanist. And he's the one that identified Quercus hinckleyi as a separate species in the 1950s. He's, let me get my... That's him lying down behind a Quercus hinckleyi. That's what it is. It's very, very small, very shrubby. And these are just some of his field notes from the University of California, Santa Barbara. The current range in the U.S. distribution of Quercus hinckleyi is limited to one county in Texas. There's one sighting that's just right across the border into another county, but essentially is this one county down here. For those not from the United States, you're right up here near Chicago. And of course, there is another country here called Mexico. That's just not shown on this particular image. So, as the climate changed after the last ice age here, the area where hinckleyi is found has become more and more dry. About 12,000 to 4,000 years ago, it was much more wet, and actually there have been fossil Quercus hinckleyi found in pack rat mittens that show its range was much wider than it is now. But as time went on and it dried out, now it's very, very rare. We sampled pretty much everything that's known in this particular, in the United States, and we ended up with 123 different plants. So very, very rare. And this is its habitat. This is Chihuahuan desert habitat, which is typical of where it's found. This down here is actually a geologic formation in Big Bend Ranch National Park. And if you can see these little indicators here, that's where our samples were taken. Some of our samples were taken. So it's pretty rugged, pretty isolated. It's found on limestone generally, some sandstone, and elevations of 1,000 to 1,400 meters. And it grows in clumps. As you can see, what Muller was sitting behind is these little clumps, and those are the height of Quercus hinckleyi, less than a meter. Reproduces clonally through rhizomes as well as sexually through acorns. And the clumps can vary in diameter from less than a meter to kind of a wider group like this. The leaves, this is a mature leaf, this is it. And they're gray-green, they're adapted to the xeric climate that it lives in, they're thickened, they're holly-like, very, very small. So when you're working with them in the lab, if you sneeze, you might lose all your samples. It was challenging at times. So conservation issues, again, it's a very, very rare species currently. And it's listed as threatened under the U.S. Endangered Species Act. It's listed as threatened by the state of Texas and the IUCN Red List. Some of you were at the meeting on Sunday. It's critically threatened. But the U.S. Fish and Wildlife Service has developed a recovery plan for it in 1992, which was reviewed in 2009. And within that plan were 3 priority 1 tasks that were that could be answered by genetic research. So that's where my genetic research came into it. So the first one is to just assess the genetic variability of it. Second one was to look for types of reproduction. And the third one was to see if it was being threatened by hybridization, by being swamped by other oak species. So here's the genetic study. I'm not going to go into a lot of details. You're going to see some tedious looking tables, but we won't go through all the numbers, so don't worry about that. But to give you an idea of what we go through, we use microsats that Vicki had talked about. It starts off by collecting the samples in the field. Those come back to the lab. We extract DNA. Then we do the microsat analysis. And then from that, we determine what the alleles are within the plant, within each plant, at each locus. A locus is just like a little area of the DNA. Run that through then various types of analyses. And so it's really here where we determine what alleles are in a particular plant. And there'll be two alleles, one from the mother, one from the father. They could be the same length, they could be different lengths. But by analyzing that, we can look at the diversity, and we can see if they're unique individuals or clones, and we can look for introgression and hybridization. So that's our key finding here. So let me kind of look around the corner here. These are our sampling sites. So Hinckleyi is just found right in here. We had 4 different sites that we sampled. 2 are near the town of Shafter, Texas, which is officially a ghost town, but actually there are people living there. One of those is on private land and one is on mining land. So there's a little bit, private land is kind of monitored by the residents that are near there. Mining land is, of course, in a restricted area. And this area down here is Big Bend Ranch State Park. And those are the other locations of our sites. And it's about 60 kilometers between those two. So to look for the hybridization potential, the literature told us that pungens and Vassiana were potential hybridizers. So we got some pure pungens and hybridizers up from Guadalupe Mountains National Park, which is about 320 kilometers from where the Hinckleyi is found. And then we found a stand of pungens that was close to one of the stands of Hinckleyi, very close. Actually, the Hinckleyi are in the drip line of the pungens. And then there were 2 trees that people thought might be hybrids, and those were near the Shafter sites. So we also looked at those to see genetically what physically people thought might be hybrids, if genetically it was confirmed or not. So don't worry about the table, but that's the kind of information that we get from our analysis. And we're looking at numbers of alleles and various other statistical numbers in there. But what you need to come away with is that we found genetic diversity was high. Even in the small sites, we found it was high. And I'll kind of talk a little bit more about that, because those small sites were very much clonally represented. And then we found no evidence of inbreeding. So there was enough sexual reproduction going on, and we also think that the clones were actually maintaining enough variety, and were not able to sexually reproduce to cause inbreeding. So some interesting findings there. But in essence, we answered the question about genetic variability that was in the recovery plan. The second question in the recovery plan had to do with reproduction, what kind of reproduction was taking place. And these are the 2 small sites near Shafter. And we sampled 58 ramets there. And the clumps were separated, and we thought, well, we'd have quite a few individuals. Actually, after the genetic analysis, there were only 7 unique individuals amongst those 2 sites. In the Big Bend Ranch State Park sites, less cloning, more sexual reproduction. But as you can see, the total of ramets that we sampled was 204. After genetic analysis, there were only 123 unique individuals. I know we've had discussions of how do you count these things, but being kind of biased towards genetics, I guess, you really know which ones are unique, or have to know which ones are unique to get a real valuation of what you have. So I'll give you an idea. This is one of the small sites, the S1 sites that had a lot of cloning. And we came in, and the flags would be different clumps, different discrete clumps that we sampled. And again, we thought we would get quite a few unique individuals. And actually, the ovals there are really the unique individuals. So there were 4 out of all this. And in fact, this one was about 30 meters across from one clump to another clump with nothing in between. So what happened, I feel, and I'm pretty sure, is that it cloned from a central area that died out over time, and you kind of see this ring of clones that are left. And then another thing we look at, and maybe I can talk a little bit about this, it's another tool that we use. We put the data that we analyze into this tool. And what we're trying to find is within all of our population, are there any like clusters that are the same, genetic clusters that are the same? So subpopulations. So you don't see it too well here, but what this picture is showing you is there's a column for each of the individuals that we tested. So for example, this one has like maybe an F1, maybe not, a mixture of these 2 different genetic clusters. These are all Hinckley oaks, but their genetic material is very similar for these 2 and then for this one here in the middle. And the significant thing about this, if you go back and look where we got these, this is again the Hinckley sites that we sampled. This one, which is kind of orange in this picture, came from up here. This one, which is its mate, came from 60 kilometers away. And in between, it was this other cluster. So you're going like, okay, what are we seeing here? And remember back that it had a much broader range at one time, which has slowly diminished as the number of plants has diminished. And so our feeling is that that similar genetic cluster probably connected those at one time, but has been lost over time in between. Other possibility is this is very rough terrain, and maybe we'll find a valley with some Hinckley in it, hopefully. Then the last one that we looked at, The last one that we question that we looked at was hybridization, and there's the Hinckley again, and the putative hybridizers that we picked up from the literature, Quercus pungens and Quercus bastiana, and as you can see, the leaf structure is quite different, so Hinckley really stands out in its native area. And without going into too much of this, we said that if we found Hinckley that had less than 85% of the Hinckley cluster, it would be considered a hybrid. And so we found very little, actually. There was only 5% of everything we worked on, we sampled. And so we found no evidence of genetic swamping. And our thinking on this is that it's so isolated now, and there's so few other oak species nearby that that's helping it from being genetically swamped by other species. Of interest was kind of a reverse introgression that we found. This was a group of Quercus pungens that looked like Quercus pungens. They had no characteristics, of Hinckleyi, and they were located directly next to a relict Hinckleyi group. Actually, the Hinckley was on the drip line of this stand. And we found a kind of a reverse introgression. So these plants had a lot of Hinckley in them, although they showed no physical evidence of it. And so that, again, was kind of an interesting thing. And it seems like they are actually acting as a repository for some of the genetic material of Hinckleyi, which kind of fits in with what everybody else is saying here too, but also from a conservation standpoint, it makes you consider protecting habitat, not just a species, because there are things going on that you don't realize. And then the two hybrids. The first one here, my colleague in Texas had written a paper where he looked at the morphology of this particular tree, which again was near the shafter sites. As you can see, it's a tree form. But what he found in it was leaves that were small like the Hinckley and then more like the Vassiana species. And so he wrote the paper just based on morphology. We didn't know, there was no genetic analysis involved. But we looked at the genetics of it, and in fact, it was like 50-50 between those two. So that one definitely a hybrid. The other one, again, a tree form. You'll recognize this person, the next speaker, to give you an idea how big that was. Very close to one of the relic sites. The leaves, not particularly Hinckley, but it was so close that we considered there be introgression into this, and there was none. It was perfect. It was totally the Vassiana, the pungens genetic material. So then, that kind of answered the conservation questions, but we wanted to look at this in kind of a larger context, and there are assumptions about rare isolated plant species. So I want to go through those assumptions and how Quercus either contradicted those or followed what they were. So we'll start with, the first one was that there are low levels of genetic variability. And we found in the Quercus hinckleyi that we sampled that it was comparable, the variability was comparable to other oak species that are non-threatened. So it's high. There's an assumption that there'll be a lot of inbreeding. And we found no evidence of inbreeding. So we had that one area where there was sexual reproduction, and there was an area that was very clonal. And the thinking is that the clonal area, because it is not reproducing sexually, and there are so few individuals, actually if it were reproducing sexually, there would have been inbreeding. So it's being protected by cloning, which is kind of maybe not a common way of looking at cloning. And then there's the assumption that there'll be limited genetic sharing between separated populations, and as you saw, we had that one widely separated subpopulation that was 60 kilometers apart. So that was contradicting that particular assumption. And we feel that that's remnants of more widespread historic distribution. And then there's the assumption that species identity can be threatened by hybridization. And in Quercus hinckleyi, we just didn't find that. And it could be because of the isolation that the remaining plants exist in. But we did find that congenitor species, that Quercus pungens stand, that seemed to be a repository for some of the threatened species genetic variability, which is interesting. And then the assumption that cloning limits the number of unique genetic individuals. Obviously it does. We found that. On the other hand, whoops, a little fast there. It seems to also be a means in this particular case that the clones are maintaining genetic diversity. And then finally, the assumption that limited numbers make them especially sensitive to habitat degradation and climatic effects. And everything else, the hinckleyi that we studied contradicted the assumptions, but not this one. This is definitely yes. It's threatened. There are human effects that are threatening it. They're talking about a natural gas pipeline going through some of its territory, very close to those shafter sites. Bighorn sheep are being introduced into Big Bend Ranch State Park, and you don't know if they're going to have a taste for Quercus hinckleyi or not. Climate is changing, and it's probably going to get drier, and we can't control that, obviously, but that could be like the fatal blow for it. And just this habitat loss that's caused by basically human effects. And will it go extinct? I don't know. I'm having worked with this for years and years and years. It's my baby, so I'm hoping that somebody's going to find it. Maybe Harry, like a whole canyon full of it. But yeah, I mean, I don't know what's going to happen with it. So key thoughts to take away. Because we now can identify unique individuals and we know where they are. If anybody's defining a recovery plan, we can point them to unique plants rather than trying to breed clones, which is not going to work very well. So I think we've enhanced the ability to maybe make some recovery efforts. And that we found that Hinckleyi introgression into the Quercus pungens that's so close to it really kind of makes you think that you should be protecting habitat, not just species. That genetic separation by 60 kilometers is interesting because I believe we're seeing the historic range that has been lost and shrunken, and we're seeing like outliers in that. Those are not pioneers, those are just survivors. And that cloning is kind of a thing to think about, and how cloning is a positive thing, or can be a positive thing for a threatened species. And then finally, I'd like to make sure everybody understands that there continue our threats. I know you like to collect acorns, but maybe not this one. It can use everything that it's got because there's so few left. So I just pass that along. And so we can do things on a human level. I mean, we can't change the climate. We're too late for that. But if we can protect it on a human level, that would be great. And many people helped me. Many people collected for me. Mary's Lab. I can't thank enough. People in Texas, my colleague Martin Terry, Jackie Poole at Texas Parks, my husband who took pictures. So a lot of people were involved. And finally, these are the 2 scientific papers. So if anybody wants to actually get into it at that level, they can read those.